Hi, I'm Alan from CAPS here at the University of New Mexico. Today we're going to be talking a little bit about genetics. Specifically, we're going to work at DNA replication. There are a lot of videos out there detailing the whole process, so I thought we could just kind of focus in some of the really problematic areas for students today. So one of the areas that I think is most problematic for me personally and that I've seen students struggle with is just the directionality of DNA synthesis. So your mantra for all of these problems, for my mantra for these problems, which you're welcome to use, is 5 to 3 N to C. 5 to 3 N to C, because DNA and RNA are always synthesized in a 5' to 3' direction, and proteins are always made in an N terminal to a C terminal direction. So when we have our replication bubble, remember that DNA, double-stranded DNA is opened, and we have two different replication forks on either side. Each DNA strand has a direction of 5' to 3', and then RNA or DNA is going to be made on top of that in the opposite direction. So when you need to know which direction your DNA is being synthesized is being synthesized in a 5' to 3' direction, starting at the 3' direction of the template DNA. And of particular interest to us is down here in the replication fork. You could tell that one strand of DNA is being synthesized faster than the other because it's going directly into the fork. Its directionality is supported by the direction of the DNA opening. We call that the leading strand because it leads. And then the other side has this problem where the 5' end is constantly being opened and new synthesis is constantly being forced to start. And so we call this the lagging strand because it generates these short DNA segments leading to the generation of Okazaki fragments. In biology, I often think about it as being a story, and in order to know the story of the process we're looking at, it's helpful to know the characters first. So if we're interested in DNA replication, we first need to know who all is around and what they're doing. So again, we're going to start when we have double-stranded DNA opening up, and we're going to have a whole suite of enzymes and proteins coming in performing this reaction. Again, this isn't going to be too much of an in-depth process, just kind of highlighting some of the problematic areas. So one of the key enzymes is called topoisomerase. This enzyme goes upstream of the replication fork in either direction, and it helps with relaxing the DNA. So if you picture a string that's getting coiled, and then as you start pushing up on that, if you don't relieve some of that twisting, you're going to wind up with this really nasty supercoiled structure. Topoisomerase actually breaks the backbone of DNA in order to relieve that. Helicase is sort of the all-star of the show as far as opening DNA. It gets right into the fork, it's right in the action, and it's breaking those hydrogen bonds between the base pairs, and it's pushing forwards and actually opening the DNA physically. The other suite of proteins or set of proteins that's involved in opening DNA and keeping it open are called single-stranded binding proteins, or SSBPs. They bind to single-stranded DNA. That's nice because they're named for what they do, and they actually stop DNA from rebinding back to itself, so it helps keep it open. So topoisomerase relaxes the strand and lets it stay open. Helicase actually opens it, and SSBPs stop it from closing back up. Then we need to worry about how are we actually going to make new DNA. So you'll recall that DNA synthesis requires a free 3' hydroxyl group, and it requires an RNA primer. So before we can go in, we have to lay down a primer. There's an enzyme called PRIMASE that does that. Again, it's named nicely. PRIMASE doesn't require free 3' hydroxyl, so we can just lay down RNA primers anywhere. Then DNA polymerase is going to come in and do the actual synthesis and make a new strand of DNA, starting from the primer and going forwards. Primers will be degraded and turned back into DNA, and now the last step is performed by a protein called LIGASE that comes in and actually fixes these little breaks. Right? Because we have different chunks of DNA together, we actually need to stick them, we need to bind them together, fuse their backbones. Ligase is the one that does that. So it's just keeping these sets of proteins straight. You want to know topoisomerase, helicase, single-stranded binding proteins, primase, ligase, and DNA polymerase. Those are the key players. If you keep them in your mind, you can probably put the rest of the process together. Alright, so that's all we really wanted to discuss today. If there are still things that you'd like to clarify, be sure to use our online services or our in-person services. Go to caps.unm.edu to check those out, or follow us on our social media accounts. Thank you.